our second speaker today is Dr. Kristin Low, who's a postdoctoral fellow in the Agriculture and Agri-Food Canada lab in Lethbridge, working with Wade Abbott. Kristin actually worked with another GlycoNet investigator, Dr. Lynn Howell, at the Hospital for Sick Kids, and she's made the move to quite a different branch of glycoscience, and she's going to tell us today about bioprospecting for CAZymes. So Kristin, the floor is yours.
Okay, so I guess just before I start really quick, I just wanted to acknowledge and respect that I and all of my work, or at least most of our work that I'm presenting today, are on the traditional territory of the Blackfoot Confederacy. So today I'll talk to you about bioprospecting CAZymes from intestinal microbiomes. And ruminants, such as cattle and sheep, are highly adept at digesting dietary fiber, whereas monogastric animals are less so. And we can add exogenous enzymes to livestock feeds, and these have shown some great promise in increasing the digestibility of complex carbohydrates, leading to increased energy acquisition and animal performance in the agriculture industry. But in monogastric animals, the results of these enzyme additives so far have been pretty mixed. For my project I'll talk to you about today, specifically we've been looking at improving the digestion of canola meal by chickens through the development of an enzyme discovery platform that's really been tailored to specific non-starch dietary polysaccharides. Soybean meal is really commonly fed to chickens as a suitable source of protein, but canola meal is a really promising up-and-comer. When you compare canola meal and soybean meals, so canola meal, I should say, is a byproduct of the canola oil industry. And when you compare it to soybean meal, they contain similar levels of protein and amino acid profiles, but additionally, canola meal is half the price of soybean meal. So it really provides a good, or it can provide a good alternative feed ingredient, both for the canola industry and poultry farmers. Unfortunately, though, canola meal contains higher levels of dietary fibers, lignans, and non-starch polysaccharides, or NSPs, I'll refer to them from now on. And this causes really the one main problem in feeding chickens canola meal is that it's not very well digested. So what we're proposing is this approach to discover enzymes tailored to canola meal NSP digestion, and that this approach will be effective and beneficial. In the Abbott lab, we've really used this multidisciplinary innovation platform for this discovery of enzymes for the digestion of livestock feeds, which, like I said, in this case would be canola meal for chickens. We're combining a number of different expertises in our lab, microbiology, glycomics, bioinformatics, protein biochemistry, as well as combining with collaborators to do animal work. And really, just to walk you through our thought process, the idea is that initially microorganisms are collected from chickens and cultured. We enrich for these cultured microorganisms grown on all these alternative feedstocks, so in our case canola meal, in these bioreactors, to enrich for microbial species that specialize in degrading these NSPs. Alongside this, we actually do comprehensive glycomics to really characterize these polysaccharides in question to get a really full and complete understanding of what we're actually trying to degrade. From these isolations, we then do genome sequencing to identify the organism and look at its CAZyme profile. We then also do phylogenetic analyses using our SACCHARIS pipeline to generate some family trees and then help identify the function of these CAZymes. And then following this, we do some standard protein biochemistry work, recombinantly producing and purifying these enzymes, doing some characterizations, and then hopefully in the end, we can do some scale up and wind up with an enzyme product that can be then fed back into the animal models for evaluation. And using this kind of flow here, we're looking for enzymes to digest the cell wall polysaccharides of canola meal. And as a bonus, they're already used to working in the chicken digestive tract. In theory, they're already natively present at low levels. I think a lot of us here can appreciate that there's a huge diversity seen in higher plants in the polysaccharide content, as well as animal feeds, and really canola meal is no exception to this. The monosaccharide composition of canola meal had been elucidated, but up until before we started our project, we really didn't know the exact polysaccharide content and the glycosidic linkages that were contained within these NSPs. And if you're trying to discover enzymes, it's going to be quite difficult without this context involved. So that's one of the things that we really wanted to start with is, can we evaluate the polysaccharide composition of canola meal? To do this, we did a number of different approaches, one of which was using some ELISA-based screens. And the first one we started with was collaboration with Mike Hahn's group at CCRC, looking at his library of 200 plant cell wall glycan-directed monoclonal antibodies. And so this was really a broad spectrum approach. We also collaborated with Will Willitz's group at Newcastle University to go for a targeted panel of glycan-directed mAbs. And both of these, just in the interest of time, I'll say they presented results consistent with the broad panel. And to summarize, really, it showed that these canola meal NSPs contained glycans that were consistent with pectic polysaccharides, including RG1 and xyloglucan, among others. While this was really a kind of a high-level approach and a broad approach to this, we wanted to really get some high-resolution information as to what exact linkages are present. And so this is when we perform some glycosidic linkage analyses by GCMS. So we can look at all of this data, identify the linkages that are present within our NSPs. And putting this all of this information together, we can then estimate the polysaccharide abundance in the canola meal cell wall. From these GCMS analyses, we saw that really pectins like RG1, arabinan, galactan, arabinogalactan were present, along with others like xyloglucan, arabinoxylan. And this data here was really consistent with our ELISA analysis. So at this point, I think now that we know what polysaccharides we're looking specifically at trying to break down, we can kind of get back to talking about the enrichment and isolation of these bacterial species. And like I mentioned, what we did was we used these bioreactors that had a continuous flow that were inoculated with chicken cecal samples and contained liquid minimalized media that contained either canola meal or the NSPs. And these were allowed to grow and enrich over a period of 10 days. After this point, we took samples, plated these on solid media, and isolated single colonies for downstream analyses. And we managed to culture 332 different isolates from these bioreactor experiments. And I should note that 100 of these specifically were isolated from NSP plates. If we look at the family abundance for the bioreactors in their entirety, we see that although they're initially populated with largely Clostridia and Ruminococcus, over time, Bacteroides were actually selected for over the length of the enrichment. From our 100 isolates that were selected and plated on NSPs, we did some colony PCR of the 16S and showed that 2 of the predominant species with the ability to grade canola meal NSPs were Enterococcus and Bacteroides. And since the Bacteroides were selected for in the bioreactors, and we know that they're polysaccharide generalists that degrade diverse cell wall polysaccharides, this is kind of where we started our focus, was on the Bacteroides fragilis, B theta, and B ovatus. Up until this point, we had done all of these isolations on solid media, and so what we wanted to do now was identify any high efficiency degraders and evaluate their growth in liquid culture. And just to summarize all of this data quickly, I think of these 7 Bacteroides, we found that the 2 B theta and the 2 ovatus seem to be moderate degraders of these canola meal NSPs, and the fragilis really grew to low density. Moving forward with these Bacteroides isolates, we took 6 of these, including the 2 B thetas and the 2 ovatus that were more adept at growing on NSPs, and 2 out of the 3 fragilis. And we did Illumina sequencing and assembled the contigs using spades. And then these assembled contigs were run through PROCA to annotate based on putative proteins. with the output from PROCA being then fed through dbCAN so we can identify the total CAZyme content for these bacterial isolates. And as you can imagine, we got a lot of data from this, so I'll just show you a little bit of a snippet right here. And what I'll point out is that significantly these high efficiency degraders, so the B thetas and the B ovatus, seem to contain roughly double the total CAZyme content as the fragilis low degraders. You can also see that I'm sure a lot of these families, these CAZy families, were especially relevant when you look in the context of the glycosidic linkage analysis and the ELISAs, so based on the polysaccharides that were identified in canola meal. And we chose select families based on this to then move forward through with our SACCHARIS analyses. I'm sure a lot of you have heard from the Abbott lab about SACCHARIS, but just real quick, this pipeline is really developed to streamline CAZyme discovery in an automated way, whereby we input our protein sequences from our sequenced isolates, along with characterized sequences from the CAZy database in order to output phylogenetic trees for each CAZy family. And what we're looking for using SACCHARIS in terms of target selection for these enzymes is that we're looking for sequences from our isolates that are really outliers, being that they're not so closely related to other characterized family members, and these may present novel activities worth investigating. One of the families that we chose were GH28s, since they are predominantly pectin active galacturanases, and since, like I said, pectin is highly present in our canola meal NSPs, we started here and chose two enzymes that seem to have diverged from other GH28s. One of which is K2605 here was selected as really one of a distinct clade on its own, whereas N2073 was chosen as a representative sequence in this novel clade of isolate enzymes on the tree. We then also chose other families including GH43, GH78, and CE6, since these families were all relevant to the polysaccharides that were present in canola meal. And in these trees, we looked for outliers as well and wound up with a set of 8 enzyme targets to start with. Now, a number of these, as you can see, have varying associated CBMs and some auxiliary domains, and even some had unannotated space by dbCAN. So we thought this was a good mix of really interesting targets to work with. I'm sure a number of you can also appreciate that although you start with 8 sequences, when it comes to recombinant expression in E. coli, this is not always perfect. And so what we ended up with was 2 full-length sequences that we could start to screen for enzyme activity. And just to quickly walk through, what we did was we looked at 2 real assays. One was the PNP sugar conjugate assay, whereby cleavage of the PNP sugar substrate would result in the release of PNP that can be monitored photometrically over time. And the second was just a simple incubation of the enzyme with some target polysaccharides and trying to identify any product formation by TLC. And what we found, first of all, by looking at the GH43 and 1089, was that we found that it was active on both arabinofuranose and xylopyranose. And by TLC, we also saw some products consistent with oligosaccharide product formation from canola meal air. And what we then thought was really, because this enzyme is composed of 2 tandem GH43 domains, we thought that this is possibly a dual function enzyme with arabinofuranosidase and xylosidase activity. The second enzyme that we looked at was this K2605, and we really didn't see a lot of activity with this. We saw maybe some residual activity on PNP galactose, but from the GH28 SACCHARIS tree, this should be a galacturonidase. And so this really kind of, we think we haven't really targeted the true, or looked at the true substrate of K2605 yet. So just to sum up, I guess, in the end, I think we have some interesting enzyme targets to look at and further characterize, but I think it all stems from our targeted approach, that really we tailored these bioreactors to our polysaccharide in mind. We enriched and selectively grew bacterial species that were able to degrade these polysaccharide, and we ended up with some enzymes that are relevant to our linkages that are present. And I think without the knowledge of our polysaccharide composition, it makes enzyme discovery really hard. Up until this point in the past, a lot of broad spectrum shotgun approaches required high throughput assays, massive scale up, and testing a large number of enzymes in animals. And our approach that I've presented here is to streamline this discovery in an informed way, and I think this directed approach is paying off. I also want to point out that this digestibility improvement platform can be tailored at these various kind of entry points to various animals, feeds or polysaccharides, or other downstream systems. So whether this is for enzyme product development or even for just research and discovery applications, we're very open to collaborations to adapt this to different needs. And with that, I'd just like to acknowledge a number of people, specifically the Abbott lab, for allowing me to work on this project and supporting me through it. I'd also like to acknowledge Mike Zing, who is our resident chemist that does a lot of the glycomics and glycosidic linkage analyses. Doug Ingalls and his group are critical for our work. They work a lot in the chickens and set up the bioreactors to do those assays. And then a number of our different collaborators for the glycomics and a lot of the feed assembly and enzyme and canola meal approach of all of this. And with that, I'd also like to thank Alberta Agriculture and Forestry for funding my project. Thank you.